Welcome to the North Carolina Botanical Garden. Welcome to the folks on Zoom as well. I'm Joanna Lalikas. I'm the Director of Learning and Engagement here, and it's a beautiful day to be at the garden. We've got a nice hour of wonderful information to share with you, and I hope the folks here will go out and enjoy the beautiful day. And those on Zoom, come and visit us soon. It's beautiful spring. So I want to introduce in the back of the room David Michaud. He is here supporting our Zoom audience and will support Q&A from the Zoom. He's our registrar and program coordinator. Without further ado, I'd like to invite our special guest, Hannah Levenson. She's a postdoctoral research scholar with NC State's Department of Entomology and Plant Pathology. Dr. Hannah Levenson is a community ecologist interested in investigating how humans impact the environment and exploring ways to mitigate those impacts. She uses pollinators in agricultural settings with concepts of integrated pest and pollinator management as tools to research these interactions. Hannah is a bee expert with experience researching both native bees and honeybees in US and international settings. Currently is the head of the Specialty Crops Integrative Pest and Pollinator Management Laboratory. She is working closely with blackberry growers across North Carolina to make the management of an invasive fruit fly, spotted winged Drosophila, more sustainable and to better understand how management decisions may impact pollinators. Hannah, thanks so much for coming to be with us again. We had the great pleasure of working with Hannah about a year ago on a little project. So great to have her back again. Please join me in welcoming her.
Thank you for the invitation. I love coming here, working with you all, and happy to share about my research. So like Joanna said, I'll be talking about supporting pollinator communities in NC agroecosystems. So I will jump right into it and recap a little bit of the introduction that Joanna already shared. So like she said, I'm a community ecologist, and I'm broadly interested in investigating how humans impact the environment, and then figuring out ways that we can work to mitigate those impacts. But as I've been working in my research and starting some new projects, which I'll talk about at the end, I'm starting to think about more how this is really a two-way street. And as we interact with the environment and make decisions about the environment and learn about the environment, that also then impacts us, and we learn from those interactions we have. So it is a two-way street, and I'm interested in how we can utilize this back and forth interaction that's happening to improve the environment for everyone involved. Like Joanna mentioned, the main tools I use to investigate these interactions are pollinators in agricultural systems. So today I'll talk to you about some of the work I've done in North Carolina, looking for ways that we can better protect pollinators in agriculture. So I'll share a couple different projects today. The first topic is looking at conservation efforts for pollinator communities and agroecosystems. I'll talk about two projects that were part of my PhD work. The titles and information are listed here, and you can actually access the publication from this work online. If you're ever interested in more details than what I give here, you can access both of these online for free. The next topic I'll talk about is looking at the impacts of pollinator conservation on the greater agroecosystem. So as we conserve pollinators, what does that do to the agriculture system we're working in? And you can find more details again online for that project. And then the last topic I'll talk about is the development of integrative pest and pollinator management strategies in specialty crops while controlling an invasive pest. And this is research I'm currently conducting, so you'll be seeing a sneak peek of some preliminary results that aren't available yet online. So I first want to talk about North Carolina is a really interesting place to conduct research in agriculture. So this is a snapshot from northeastern North Carolina, and you can see in just this small picture how diverse the landscape is. So there's about 8 different crop types shown here, as well as there'll definitely be waterways. There's multiple types of forest. So there's a lot of different landscape types butting up against each other. The landscape can be pretty fragmented, but that also means there's a lot of little pockets in the agriculture that we can work with for conservation efforts. So that was kind of the idea for a program that really started off most of my research. It was spearheaded by the North Carolina Department of Agriculture and Consumer Services, and they wanted to work to protect pollinators in agriculture systems. So they started a program called Protecting NC Pollinators in early 2016. And this required the planting of pollinator habitat at all of the agricultural research stations across the state. So they found those little pockets on their research stations that weren't being used in other ways or were really hard to use for agricultural purposes. And they did conservation efforts for pollinators instead. And so my research visited these research stations, documented the changes in the pollinator communities over time after establishing this habitat to see what were the effects of adding this as a conservation effort. So this map shows locations of all the research stations across North Carolina. We have quite a few here. They cover basically the entire state. So it's a really interesting network to work with. I used 16 of the research stations. One is unofficial research station number 19, right next to NC State. We used 2 sampling methods during this. You can see those circled with red. Those are stations that only had bee bowl samples occurring. So this is a picture of a bee bowl down here. It is, oh, and the mouse is a little different on the screen than my computer. So these are painted small plastic cups. They are painted blue, yellow, or white. And those colors mimic the UV colors on flowers that attract pollinators. So they're attracted to these cups. They're filled with soapy water and it collects them and we can then identify them in the lab. So I had a contact at all the research stations that put these out around the plots once a month for 4 months during peak bloom so that we could document the pollinator community there. So that happened at all of the research stations. Then those circled in blue, we took an additional netting sample type, which is shown here down at the bottom. So we used hand nets. At as close to the same time as the bee bowls were placed out, we went out and used nets and collected whatever bees we found once a month for 4 months, and then put it all together to document the communities there. So I'll show you some of the results really quick. I want to orient you to this map. So again, we have the same map with the same circles denoting all the research stations across the state. We then have these colored pie charts. That represents the samples we collected at each research station specifically. The colors within the pie chart correlate to these colors up here in the upper left corner. Those are showing the most commonly occurring types of bees we found. You can see there were quite a few different types of bees. The size of each pie chart represents the relative abundance that we collected at that site compared to all the others. And we have a size key here so you can estimate the sizes that you're seeing. The other thing I want to note is along the right-hand side, we have large pie charts which show for that year, 2016, 17, and 18, all of the samples in North Carolina all put together. So on the side, we have the statewide view of what we were collecting, and then in the middle, we have each individual station. All right, so now we will look at what changed over the years. So we're looking from 2016, 17, and 18, how the abundance, the distribution of the common bees changed at each site, and then also across North Carolina. So a big takeaway that you might notice right away is that these pie charts all look pretty different. There's a lot of change going on over the years. If you pick one pie chart and follow it across the years, it looks really different. So no station looked the same as the other stations, but even the same station looked different than itself across years. But something we noticed is if you look at the North Carolina statewide along the side, those pie charts, even though the number of samples we collected, which is listed underneath, as well as the number of different kinds of bees we found did increase over time, the pie chart and the distribution of those common types stays pretty consistent. So there is this discrepancy where at the local level, we're seeing a lot of variation. But if we look at a big snapshot, the whole state of North Carolina, things are pretty consistent. So we wanted to look a little further about what could be driving this variation locally. And we found, not all the way surprisingly, but very interesting, that the habitat characteristics at that specific site were very important in driving what pollinator communities we found. So what we're seeing here is along the bottom of the charts, you have 2 measurements we took of the habitat that was planted at each research station. This first chart, we have flower cover. So we documented what percentage of the plot was in bloom at the time we collected bee samples, and that's rated as low, medium, or high flower cover. The second chart, we have a measure of flower diversity. So we documented how many different kinds of flowers were in bloom when we collected bee samples, again rated as low, medium, or high. And then on the y-axis in this chart, we're showing you bee abundance. So the number of bees that we found at each sample compared to flower cover and flower diversity. So if we look at flower cover first, we'll see that there was a significant interaction where bee abundance significantly increased as there was more flower cover. Makes sense, more flowers, more bees, but it was really great to document that that was actually happening. As these stations were adding habitat, we found more bees. So that's very promising. If we look at the flower diversity chart, we see a similar trend, but it was not significant, where we saw a trend of increasing bee abundance with increasing flower diversity. Now I want to look at similar charts. We're still looking at flower cover and flower diversity, but this time we're looking at bee richness on the y-axis, which is a measure of how many different kinds of bees we found. So again, with flower cover, we see that significant interaction. We're finding more kinds of bees with more flowers present. And this time we also have a significant interaction with flower diversity. So we see more kinds of bees with more kinds of flowers. So we were really excited to find this, and it's really promising that adding habitat does seem to be helping the bee community. But we did notice that things changed in the habitat over time. So we're still looking at flower cover and flower diversity, but this time on the y-axis, we're looking at those 4 different samples we took. And we took samples in spring, early summer, late summer, and fall. And what I want you to notice is for flower cover, in the fall, we're having more low cover. So there are less flowers blooming in the fall than in the spring. And we see a similar pattern for flower diversity. Less flower diversity in the fall than compared to the spring. So I just told you flower cover and flower diversity were both very important for supporting high numbers of bees and lots of different kinds of bees. And we're seeing that that decreases over time. So this is something for future work. If we're establishing new habitat, this is something to keep in mind. It can suggest that when we're making seed mixes, let's try to focus on having flowers that bloom later in the year so that we have more resources we're supporting for bees. We also noted anecdotally from our research stations that they had a lot of problems with invasive weeds coming into these habitats, with soil seed banks taking over the habitat. So it's just to note that this is still a man-made object that we're putting out in the environment. So it's not something you can set up and then leave and it's just going to take care of itself. We need to continuously maintain this habitat to make sure we're providing the highest quality resources for bees that we can. Otherwise, the habitat might shift into something that's no longer supporting bees or supporting the spread of invasive weeds, which is also not something that we want. All right, so we learned a little bit about what bees we found. We wanted to know what is happening at this habitat. We're attracting thousands of bees, we're attracting hundreds of different kinds of bees, but there is evidence that when bees share resources, they can also share pathogens with each other. So bees get sick just like us. And we wanted to make sure that by adding this habitat in the environment, we weren't increasing diseases that were in these bee communities. For us, it might seem like a large area of land. All of these habitats were an acre or less, but in the grand scheme of the landscape, that's a very small area, and we might be concentrating a lot of bees together. So for this, for sake of time, I won't go through as much detail, but we tested 8 different bee species and we screened them for a panel of pathogens. You can see them listed here. Just for time, I'll tell you that the top row, the top 7, are all different viruses. So these viruses are known to infect multiple different species of bees. They are known to negatively affect their health and can be economically damaging for our bee species. The bottom 2 are gut parasites. So these are also known to infect multiple species of bees and negatively affect their health. So these were very important pathogens to test for our bee community, and we wanted to see how this changed over time and across bee species. Okay, so we have another big graph. I'll break it down. It's the same graph we've been looking at of North Carolina with the different research stations circled. And then each panel represents the samples we collected at that specific research station. If we zoom into one panel, you see there's lots of components. So on the left-hand side for you, we have AM listed at the top. So that is representing all of our honeybee samples we collected. On your right-hand side, you see BI, that stands for Bombus impatiens. That is the common eastern bumblebee. So left, always honeybees, right, always a bumblebee species. And then the top of this is samples we collected in 2017. The bottom is samples we collected in 2018. If we look at just a single species box here, all of the columns are those pathogens I just introduced to you. So in the same order as I talked about them, all listed here. And then each row represents an individual bee that we tested for those pathogens. And because we were doing a certain lab analysis, we were able to quantify the infection levels. That's represented by the colors, where low infection level was yellow and high was red. So lots of information in one little graph. And I don't want you to look at the details. I want you to look at the overall pattern here. So just like before, we're not seeing a clear pattern across sites. All sites are pretty different from each other. Some sites, like this one listed here, have a lot of pathogens detected. Other sites, very few. Some years we had many pathogens detected, other years, very few. So there's not a clear pattern going on. But one thing I do want to know is that if you look and compare the left and the right-hand side, they are not the same at any site. So that is suggesting to us that this habitat, even if it's attracting a lot of bees and a lot of kinds of bees, it's not forcing them to share pathogens with each other. Yes. Not to our continent. Yeah. So yes, the patterns are showing that it suggests that this habitat is not instigating spread of pathogens between bee species, which is really great to hear. One thing I want to highlight, one of the gut parasites that we tested, it's called trypanosome. We did find it in multiple of the species we tested, but this gut parasite actually has multiple species itself. So there might be specific species that infect honeybees and specific species that infect bumblebees, but we were not able to determine that. That's something I'd like to look at in the future to see if those are things that are being spread between bee species. And then the last thing I want to know, if we look at our Bombus impatiens side, in 2017, sometimes we had low levels of infection, and then in 2018, it looks like that infection level got higher. So there's a chance that over time, as more and more bees come to this habitat, these patterns could change. So it's something we need to keep in mind. We don't want to make bees sick by adding habitat out in the environment. But as of right now, it seems like we have no evidence that we need to be concerned about this, which is great. Okay, so then moving on to the next topic, I want to bring up this snapshot again of how diverse of a landscape North Carolina has. And so once we added conservation habitat into the agriculture system, we saw that it supported bees. We wanted to know what happened at this landscape level. What happens to the agriculture? A lot of the argument for protecting pollinators is that they contribute to crop yield. So let's look at that and see what happens. A lot of this work has been done in pollinator dependent crops, so our fruits and vegetables, which makes sense. So you can see a couple examples here of researchers that have added habitat and then looked at yield in pollinator dependent crops, and they do see a benefit. Not as much work has been done on pollinator independent crops, crops that do not require pollinators to produce good yield, which also makes sense. But you can see some of the examples I have here. In recent years, there's been more conversations about just because a crop is pollinator independent doesn't mean there aren't interactions going on. They might still be attractive to pollinators, and they could benefit in some way, even if not entirely. So that led to this work where we looked at the impact of adding pollinator habitat into soybean fields. Soybean is one of the most commonly grown crops across our planet. In North Carolina, we grow millions of acres of soybean every year. So it's a huge part of our landscape, lots of opportunities for pollinators to interact with this crop and this crop to interact with pollinators. So to study this, we used the same research stations we've been talking about. We focused on 8. You can see those documented in the blue circles there. And then at each research station, we took bee samples in 3 fields. The same habitat we've been going to since 2016. This project happened in 2019, so the habitat was 4 years old at this point. We then sampled at the square that's labeled adjacent. So that was a soybean field planted as close as possible to the habitat. That acted as our treatment field. It should experience effects of having habitat added into the environment. Then we have our negative control, which is a soybean field planted as far away as possible on each station to the habitat, and it should not experience any direct benefits of having the habitat planted. So that was our experimental setup. So to then collect the data, we did bee surveys. From the bees we collected, we then collected pollen off of their bodies and identified it under the microscope to see what plant type that pollen was from. And then we took collections from the soybeans themselves to get some yield measurements. So first, I'll talk about our bee results. So what we're seeing here, the first bar is the habitat that we have been going to. Then we have that adjacent soybean field that's acting as the treatment field, and the negative control soybean field that should not experience any benefits. And again, we have the commonly occurring bees shown on the side, this time with patterns instead of colors. So the first thing I want to note is that in the adjacent field, we found multiple hundreds of bees in that soybean field, and then as well as the negative control field. So for a crop that's considered not pollinator dependent, we were finding quite a lot of pollinators in that crop. So that was exciting already. The next thing is there were bee species that were more commonly found in the habitat. You can see that boxed in red there. And they were less common in the soybean field. So having this habitat next to a crop field is not going to instigate a pollinator species to pollinate a crop if that's not a plant it's already inclined to go to. So it's not changing pollinator behaviors. But we did find that some of the major crop visiting species were also found in the habitat. So that circle or that box there is showing you honeybees at the top pattern and bumblebees at the bottom pattern. So this could suggest that this habitat is serving as a very important supporting resource. A lot of crop species have very short bloom times, and then what does the pollinator do when that crop is no longer flowering? So having habitat nearby adds resources for those bees into the landscape and can help them pollinate the crops better. So to see what bees we're collecting, we then did the pollen identification under the microscope. We have another big graph, but don't worry, I'll walk you through it. So the first column, honeybees, Apis mellifera, our friend we've been looking at. The middle column, bombas impatiens, another friend we've been looking at, the common eastern bumblebee. And then the far column is Xylocopa virginica, which is a carpenter bee. We then show the different habitat or field type that bee was collected in. So along the bottom, for each of those bee species, we have habitat, adjacent crop, negative control crop, the same fields we've been looking at. And then we broke down the pollen we identified into 3 types. So the top row is soy pollen. So if that bee had soybean pollen on it. The middle row is habitat pollen. So any of the flower types that we knew were planted in the pollinator habitat, we categorized into that category. And then the bottom row is environmental pollen. So anything that was not soybean pollen, not from the habitat, that includes wheat, corn, tree pollen, also clover. There was lots of white clover around the farm that was not planted in the habitat. So any of that went into environmental pollen. All right, and we found a few interesting patterns. So first, all the bees we looked at that were collected in the soybean fields had soybean pollen on them. So bees are visiting soybean flowers and they are collecting resources from those flowers. So this was very interesting to find. But there was a significant interaction between the specific bee species and the field it was collected in. So different pollinators are having different foraging behaviors, that is known, and that's still happening in this system here. Related to that, a little bit related to the pollinator foraging behavior, but we found that bees that were collected in the adjacent soybean fields, that field planted as close to the habitat, had more habitat pollen on them. So that's suggesting they're moving between fields, and it further suggests that is an important supporting resource for those bees that are pollinating our crops. They are also going elsewhere and getting further nutrition. And further supporting that this habitat helps those bees is that those that we collected in the negative control field had more environmental pollen on them. So when bees were in soybean fields planted further away from the habitat, they were still going out and getting other resources. They were trying to find trees, they were going to other crops. So it suggests that if you have a crop you want good pollination for, it would benefit you as well as those bees to provide habitat as close to that crop as possible. The bees are going to go find resources elsewhere no matter what, so you might as well give it to them right next to where you want pollination to happen. So this can be used to help improve soybean yield. For sake of time, I'll just tell you, we did find that some yield measurements were improved in the adjacent soybean fields. So beans collected in those fields were about 7% heavier than compared to the negative control field. So it also suggests we might want to rethink how we manage these non-pollinator dependent crops, because whatever management we're having in those crops is still affecting pollinators, even if we're not expecting them to be there. And even if total yield is still okay without pollination, there are ways to improve that yield. So let's look at how we can make this whole system work together. All right, so for that, I just wanted to show a couple fun examples. We collected a lot of data for those projects, and we want to get the most information out of those bees as possible. So bee identification, there is a big steep learning curve that I had to go through. And we used that information to create this pollinator identification guide, The Bees of North Carolina. If you search that title, it's free as a PDF online through NC State Extension. I wrote this with Elsa Youngstead, a professor at NC State. And it went along with an in-person day-long identification workshop that was put on pause during the pandemic, and we are starting that up soon, so very exciting. We're also looking at other measures of bee health. So here I'm showing a project I'm working on with an undergraduate at UNC Pembroke, and bees have mites on them, and different mite species are positive and others are negative. So we are now documenting what bees we collected had mites on them, how that relates to the pollinator habitat characteristics I talked about, and how can we use that to create better pollinator habitat in the future. And then the last project I'll talk about is also with another undergrad, Ren Rooney. And we are using that pollen identification that we did under the microscope to document what plants in the pollinator habitat are bees actually utilizing. So these were commercially available seed mixes. They weren't specially made for North Carolina. So there's a chance that we could have better mixes with better species added in. So by understanding what plants pollinators actually visited, hopefully that can help improve seed mixes in the future. All right, so now we'll move on to the current work. The work I'll share, I want to say at the beginning, is part of a national grant. There's about 10 different universities and several USDA offices all working together to improve the management of that invasive fruit fly we mentioned, spotted wing Drosophila. So just to quickly introduce the pest so you understand the system we're working in, spotted wing Drosophila gets its name from the male flies. On your right in that image, they have those very distinctive spots on their wings. So that's how they get their name. They're originally from Southeast Asia. They were first detected in the US and California in 2008. And then in 2 short years, by 2010, they were found all the way across the country in North Carolina. So they have very rapid reproduction, multiple generations per crop type. So they have multiple generations across the entire season. And they're very difficult to control for several reasons, but one of which is they have a very wide range of hosts that they can use. So they have more than 30 different crop and non-crop hosts. To show you some of those, they can utilize multiple types of flowers to lay their eggs in, even mushrooms, even manure, even compost. So this is a huge agricultural pest, but they're also living in our urban areas, in our home gardens. They're basically ubiquitous in the landscape at this point. And what that means is that they have something that they can utilize at all times in the year. So in the winter and early spring, before there's a lot of flowers out, they can use the decaying fruit, the mushrooms, the manure. And then as soon as the summer season starts up, they have lots of options of crop and non-crop hosts. And when we're talking about crops, these are some of the main crops that they affect. So this is data from Western North Carolina. And you can see that across the whole season, as fruits that are being grown are shifting, spotted wing can shift and utilize all of these different fruits. So all of these small fruits and berries are known to have big issues with controlling spotted wing Drosophila. Most of my work is done in blueberries, and you can see at the end of the season, based on the timing of the year as well as the structure of the berry, blueberries are very vulnerable to spotted wing infestation, and their populations are in the hundreds each week in these berries. One of the reasons spotted wing is a very hard pest is because the females have this very large ovipositor. That's how they lay their egg. And if you compare that to other fruit fly species we have here, you can see that it's much smaller than the spotted wing female's ovipositor. If we were to zoom in on this, This is her ovipositor. It's very large, sclerotized, it's hard, and it has those serrated teeth that you can see. And what that means is that they can cut into completely sound fruit that's harvestable, sellable, and lay their eggs. So in that circle, it might be hard to see in this light, but that is a spotted wing egg that was laid just under the surface of the fruit. And this is what the eggs look like if you were to remove them. Once the eggs hatch into a larva, they then eat the fruit from the inside out, which means that growers can't sell this fruit. Right now, there is a zero tolerance policy for spotted wing infestation. So if a grower has a shipment of fruit and a single larva is detected, that marketer could reject the entire shipment. So it's very costly to growers. Consumers don't want to see this, but that means that there's lots of use of insecticides to try to control this pest. So these are the insecticides that are registered for use in spotted wing. The ones that are highlighted in purple are registered for use in caneberries, which includes blackberries and raspberries, which I work in. But what you'll notice is this dotted line shows the level of efficient control we want, and that reduces the number of pesticides that are available to growers greatly. And of further concern, some of the most commonly used pesticides, there's evidence of pesticide resistance forming. So this is example data from California strawberries in 2020, and spinosad and Mustang Max are two very commonly used insecticides, and they have documented resistance. So that is a really scary thing. Our project group is currently working to document populations across the country to see if there's more resistance forming. Right now, we have found it only in California, not in North Carolina, but it's definitely something to be concerned about. So what led to the research I'm about to talk about is these concerns of insecticide use. So prior to spotted wing Drosophila showing up in North Carolina, our blackberry growers reported spraying once or twice a season. Now they make weekly insecticide sprays. Depending on the variety, that can be as much as 20 sprays per season. So from 2 to 20, pretty drastic. And even with that, the growers I work with at the end of the season, they have larvae in their berries. There are berries that are not usable at the end of the season with weekly sprays. So it's a concern economically for the growers. It's a concern for worker exposure. It's also environmental concern. And there's more pests that are now a problem. So this high use of insecticides affects multiple insects. There were lots of beneficial insects in this system that are now being affected. So there's secondary pests like mites and aphids that weren't a problem previously that are now a problem because their natural enemies are being affected. And of course, we have a concern about pollinators. These are berries, they require pollination or at least benefit from pollination. And we have no idea really, there's some information, but not a lot, how this increase in pesticide use is affecting the pollinator communities here. But there's still the concern that what do we do when there's these larvae in the berries and the growers can't sell them, customers can't eat them, what do we do with that? So we're trying to figure out how this all works together. So the project I'll talk to you about uses blackberries in North Carolina. Some of our varieties have open flowers that have pollinators visiting them. At the same time, on the same bush, they have ripe berries that require management for spotted wing Drosophila. So there's this conflicting need of wanting to protect some insects, but needing to manage others. So we went to 4 different sites across North Carolina. We collected samples along a field edge that was adjacent to a forest edge. So I showed you all those alternative hosts that spotted wing can utilize to lay their eggs. So we know that they are reproducing in the natural areas, but we also know that natural areas are great resources for pollinators. So we sampled at an edge that was next to a forested area, and then we sampled in the interior of the field, which is removed from that natural area, to compare how things change across the field. We took multiple data points, we collected berries in the field, and we screened them for counting how many spotted wing larvae were in the berries. If you're interested, I can get into more detail about how we do that, but for now, I'll leave it at that. We took pollinator surveys when the blackberries were blooming to see what insects were visiting these flowers. And then when the berries were ripe, we collected berries from the same locations, and we took a bunch of quality measurements, including length, width, weight, symmetry. And what we have found so far, so like I mentioned, this is preliminary results. We still have quite a bit of work to do. But what we found so far is we found a very diverse community visiting blackberry flowers. We were very excited to find this. We found about 24 different insects visiting blackberries. And you can see that includes flies, butterflies, and moths, beetles. But about half of those species were bees, and you can see from the pie chart, more than half of the abundance was also bees. So bees were all over these blackberry flowers. Some of the species we found bumblebee species, multiple different kinds of bumblebees. Sorry, still remembering the mouse looks different up there. And then we also found honeybees. Some of our growers do stock honeybees for pollination. But we were excited to find some native bees that are less commonly talked about. So this first picture here is the small carpenter bee, or Seratina, and we found multiple species of leafcutter bees in these flowers as well. So this picture shows one of our blackberry farms, and you can see that the bushes get quite unruly across the season. These bushes continuously grow across the whole season. So it's very common practice for growers to trim it back so that it's easier to manage, easier to navigate through the fields, but it's also easier to harvest the berries when they're ripe. And we found something interesting where these branches, or canes as they're called, when they're cut back the previous season, these old canes had these holes at the top. So we were curious what was happening. We took a clipping of them, brought them back to the lab, split them open, and we found multiple species of bees building nests in these blackberry bushes. And we found that this first nest, we believe based on time of the year and structure of the nest that it's this leafcutter bee. Right now we're rearing them in the incubator to see if we can get some adults to identify. And we found some adults, you can see one here in this nest. So we know that this is the Seratina, the small carpenter bee I shared about. So the bees that are visiting the flowers, adding to pollination, are living within these blackberry bushes, living in the canes. And this is something the growers were not aware about. So I want to share some of the berry results we had since I brought them up. They are preliminary, but we are finding differences comparing the edge to the interior, both in berry quality as well as the insect community. So we were finding higher abundances of bees at the edge of the field, but more diverse communities at the interior. We were finding that the berry symmetry, as far as how many drupelets were formed on the berries, were different at the edge and the interior. So this is something we want to look at further, especially since it's also a concern for spotted wing, these same areas in the field. We also found, though, that the variety of blackberry that we were working in had a big impact on some of our results. So we're currently working on how to disentangle the effects of the environment, the landscape that these plants are in and these bees are in, versus the crop variety being grown. So that's something we're still working on. And I want to bring it back to enabling IPPM, so integrative pest and pollinator management. So one really great example is these growers regularly go through and trim their bushes, and that just happened to create this great habitat for the bees. So now that we know this is happening and it's something they're already doing, how can we improve this? How do they trim back in a way to provide the best nesting habitat for these bees? So let's work together and figure out little changes we can make to something you're already doing that can improve your pollination. The other thing you might be thinking is I talked about how much insecticide input has increased in this system, and these bees are living in these bushes, and we're not sure how that impacts their development and their life cycle. So that's something we want to explore further in the future. Right now, we have no idea what's going on with that. So definitely more questions to be had, but very exciting results so far. Okay. So I have a ton of people to thank, and then hopefully I saved a lot of time for discussion and questions. So these are for the first projects I talked about, going to the habitat, testing for pathogens, looking at the soybean. I want to thank everyone at the research stations. They planted the habitat, they maintained it over time. They also helped me with the bee bowl sampling. They did a ton of work on this, and we couldn't have done it without them. Also, all of my colleagues, committee members, lab members, and undergraduates that helped me collect samples in the field, process samples in the lab, identify bees, and then all of my funding sources along the bottom for those projects. And then for my current work in blackberries, I want to thank all of the project collaborators across the country for doing this work together on spotted wing. I want to thank my growers and farm managers for helping me and allowing me to do this work on their farms, and all of the undergraduates that helped me collect samples and process them, and then my funding source, of course. All right, so thank you again for the invitation and letting me share my research, and I'd be happy to take as many questions as we have.
Great, thank you, Hannah. That was fantastic. Please give Hannah a hand. So I'll ask our in-person audience first. Do y'all have any questions for Hannah? Yes.
So what flowers were they planting in the habitat, and where did they get if it was a seed mix, did they get it from North Carolina or from somewhere else?
Yes, thank you. So that was a commercially available seed mix. It's from American Meadows. You can go online and buy the seed mix yourself. They tried to focus on the southeastern wildflower seed mixes, but that was not a North Carolina specific seed mix. There were definitely plants in there that were not native to this area. Since then, the NCDA has started focusing on sourcing their seeds more locally and trying to find local varieties to plant. So that's still a work in progress, but when I did the work, it was just a general seed mix.
I know some folks who have native plant seeds. How about other questions in the room?
How are you managing the habitats from year to year? You mentioned invasive plants.
Yes, so you're talking about at the research stations? Yeah, so the research stations are leading that part of it. We didn't have a lot of say in where it was planted, what was planted, how it was managed, because it was their own program, the Protecting NC Pollinators. We were just going in and documenting since it was happening, but the setup of the habitat was not part of my research. With that being said, though, I will tell you that the seed mix that they were using on the package is advertised to last for up to, I think it said 2 years, 2 to 3 years. So at the beginning, they were following the package. They were waiting a couple of years to reseed. They were just letting the habitat do whatever in the meantime. And that's how we were able to document those changes that happened over time, and that the soil seed bank was overtaking the pollinator habitat because they were following label instructions. So we have recommended to them since then to not follow that and to continuously manage every year, so that they are getting the seeds they put in the ground back out as flowers. As time went on, in addition to whatever was in the seed bank, there's also grasses. There's almost always grasses involved in those pollinator mixes, since a lot of butterfly species need grasses to lay their eggs. But grasses grow very fast and can also take over the flower side of the habitat. So as far as that goes, setting up the habitat, they tilled the area, put in the seeds, but all of that was done by the research stations.
Any other questions in the room?
Is that the,
Hang on, Ed. Here.
okay, just quickly, is that the research station south of Asheville?
Yeah,
Yeah,
this is the
okay.
Waynesville one.
The other one I've been, Waynesville, okay.
Yeah.
Asheville, okay, thank you.
David, any questions online? Got another one here. I'm coming to you with the microphone.
So, so to drill this down to my backyard, what do you have any suggestions for the best kinds of flowers, plants to plant in your own garden, in your backyard, to encourage pollinators?
Thank you. Yeah, so based on my research, I don't have a magical seed list to tell you. That's what we all want, right? There's a lot of others doing work on that. Debbie Roos has a lot of really great resources for North Carolina plants. So I go to her when I have questions, honestly. But I will say that from this, what we found is any habitat is better than nothing. Even if it's very small, it's better than having no flowers. So doing whatever you can with what you have is a great start, and then you can always build out later. I will tell you some of the most commonly used flowers that I remember. Cosmos were always very popular. Blanket flower was always very popular. Sunflowers. Zinnias were pretty popular. I noticed more with butterflies and moths than bees, but those are great pollinators we want to support as well. So those were the big ones in the habitat that I remember being popular, but hopefully with this project, let me go back to the slide, that Ren is doing, hopefully we can get some more answers specifically about this habitat.
Just from personal experience, because we love to see the bees and the pollinators, we've used portulaca and purslane, which are really good southern tolerant plants. You don't have to water them. You can go on vacation for 2 weeks and they're fine. And they close at night. But during the day, the bees and butterflies went crazy. It was like catnip. So if you want something to come there and just be pretty and also pollinate, we had them all, bumblebees, honeybees, butterflies, all over the place.
Thank you.
In terms of resources, we also have a pollinator toolkit on the North Carolina Botanical Garden website as a resource. Chiquita, hello.
So how were the habitats interspersed with the, I guess, the crops? Are they on the outside like a hedgerow, or are they within the like crop beds, like crop native plants or anything of the sort?
Yes, good question. It differed from station to station. I'll pull up this though. So in here, the habitat was surrounded by crops, almost completely surrounded by crops. The other thing I didn't bring up because we didn't document it in my research, but the stations planted more than one patch of habitat. Some of them had one large one, some had as many as like 8 different patches across the research station, depending on the year. So they were really finding whatever spare area they had to plant this. And I'll go back to the picture of the Waynesville research station. For this station, I don't know if it's shown, but their habitat, for example, would be along an edge. So it wasn't next to a crop field. So it varied a lot. And I don't have anything to say as far as like the habitat community, other than in the soybeans, we did see having it near crop helped the crop. But as far as the habitat goes, it's more important to have the habitat wherever you can, in my opinion.
I have a quick question, if you don't mind.
Yeah.
And raise your hand if you have another one in the room. I'm curious on the spotted winged Drosophila. Have you or do you plan to do research on habitat impacts on perhaps predator insects that might reduce populations of spotted wings?
Yeah, that's a great answer. It's something I think more in the future, but it's definitely a question I'm very interested in, especially since we documented how many pollinators are visiting these flowers. I'd be interested to do something similar to the soybean project, where if we add habitat, what do we see happens? But I also want to note that right now there aren't really effective predators for spotted wing here in North America. The biggest predator for spotted wing in its native range is a parasitoid wasp. And recently our project group started releasing those wasps here. It was approved, permitted by USDA APHIS. It was a long process. We didn't just go out and do that. So we're currently seeing how that impacts spotted wing populations. And in doing that research, we've been finding other parasitoids that have come in because they parasitize spotted wing Drosophila. So the predators of spotted wing are now coming into the US, but we didn't have very effective native parasitoids here for spotted wing. So more information to come in the future, but we're hoping that all of the alternative hosts in the natural areas, like mushrooms and manure and all the decaying fruit at the end of the season, we're hoping that those parasitoids that are coming in can help control spotted wing when there's not management and crops happening. Right now, there's no control for them in the off season, so that would be great.
Oh, got one question in the front here, excuse me.
I recently heard that we're not supposed to mow until the end of May because of pollinators, but I wondered, does that depend on where you live? Because it would be different in the north and in the south. And
Yes, that is a great question. I didn't share it today, but with Elsa Youngstead, who's a professor at NC State, we are currently doing a more citizen science project. It's with master gardeners across North Carolina and multiple counties. And we are currently trying to answer that question further because there's a lot of information out there, but sometimes it contradicts itself and most of it is anecdotal. And what if that doesn't apply to your region? So very good question. We have the same question. So this is our second year where master gardeners are collecting stems at different time points across the year, sending them to us, and we're seeing when and where bees are nesting in those stems. And as of right now, we have found that the summer, of course, is very active for nests and the winter is not active for nests. And we're trying to narrow down to give a better recommendation for what time of year to cut back. So great question. I don't have a perfect answer for you yet, but I agree that it would change depending on the region that you're in.
Okay. I'll ask you one more question.
Okay.
So with the habitat project, you looked at the bees and the native bees, and thank you for including the native bees in the research as well. Anybody doing research on habitat and other types of insects in crops that have benefit to the crops, either predator insects or, beyond the pollinators?
Yes, great question. There are some project groups looking at that, and there's been differing results. I think it might more depend on the crop you're looking at and the region you're in. There's a lot of location variation going on, but people have looked at natural enemies and found that that improves. But there are also some pests that can utilize the habitat, which would then negatively impact the crop. So I think that there isn't going to be one solution. And that's something I also should have said about our habitat research is we found different bee species responded slightly differently. And of course, other insects would also respond differently. So it should be thought of as a case-by-case basis. What do you want out of the habitat? What specific situation are you adding it into? What crop are you looking at? And then adapt to that situation. There's not going to be one answer that can blanket help every problem that we have.
Thank you.
Yeah.
Any final questions? Okay, Hannah, thank you again so much. We're so grateful to have you here at the garden and look forward to more of your research to come.
Thank you.